So we'll start today with disease and immunity. And the first thing we want to talk about is Edward Jenner's experiment. So he injected material from a cowpox sore into the arm of a healthy boy. So cowpox, you would get sick, but not as sick as if you got smallpox. So what ended up happening is the cowpox injection conferred some immunity to smallpox. So after the boy had healed, 6 weeks later, he injected material from smallpox into the boy's arm, and the boy remained healthy. So this child was his son, and the French called Jenner's procedure vaccination, which means encowment, right? Because it used cowpox. Louis Pasteur developed immunization procedures for other pathogens, called them vaccination in Edward Jenner's honor, and we still use that to this day. All right, Robert Koch, first to link a specific pathogen to a specific disease, injected blood from anthrax-infected animals into healthy animals, and subject animals developed anthrax and had bacteria in the blood. And when these bacteria were grown in a lab and then injected, they caused anthrax. So he isolated anthrax and determined that the sickness caused by it and the pathogen were linked together. So your body has 3 lines of defense: barriers at the body surface, non-specific responses, and immune responses. So let's talk about the first one. Barriers at the body surface are intact skin and mucous membranes, normal bacteria flora, so you normally have bacteria on your skin and your body, and then flushing effect and low pH of urine. Okay, so these are normal barriers. So you flush things out, you have normal bacteria that block, and then skin and mucous membranes help to prevent things. So these are just general barriers. Non-specific responses: lymph nodes trap and kill pathogens, regardless of which pathogen it is. And natural killer cells attack a range of targets. And then we have inflammation. So all of these are going to be your body's response regardless of what it is. Okay, regardless of what it is. Complement system: plasma proteins take part in both specific and non-specific response. Activation in one triggers a cascade of reactions that activate the others. Okay, so they activate the others and result in the destruction of the pathogen. So acute inflammation: non-specific response to foreign invasion, tissue damage, or both. Destroys invaders, removes debris, and prepares the area for healing. And characterized by redness, swelling, warmth, and pain. Okay, so we see inflammation occur depending on what has happened. So you could have some kind of infection, you have acute inflammation, and that helps, all right, destroy the invaders. So we see inflammation here. Mast cells release histamine. Capillaries dilate and leak. Complement proteins attack bacteria. And lastly, white cells attack invaders and clean everything up. So that's what inflammation does. Now we're going to get into specific immune responses. Directed against specific invaders, carried out by T cells, B cells, and macrophages. Communication signals such as interleukins play a vital role. All right, now let's look at the features of the immune system. Immunological specificity: B and T cells zero in on certain kinds of pathogens. Response is pathogen specific. So you have the B and T cells zero in on certain kinds of pathogens. So they attack specifically. Immunological memory: immune system recognizes and reacts swiftly to a pathogen it has seen before. Memory and effector cells: when B or T cells is stimulated to divide, it produces more than one cell type. Memory cells are set aside for future use. They are the basis for immune memory. And effector cells engage and destroy the current threat. Okay, so remember, memory cells are why vaccines work, why you don't get the same disease multiple times. Key components: MHC markers, antigen-presenting cells, helper T cells, effector cytotoxic T cells, natural killer cells, and B cells. So let's look at the overview of interactions here. So we have the antibody-mediated response. So if you can see at that top one, you see that there's a response by a B cell, and then the helper T cell, and then the antigen presenting cell. So you can see how they all move within each other and how all of these are going to work together. But you see that the antibody-mediated response, your body produces antibody. We're going to get over let's go over those a little bit more in depth without this diagram. So let's talk about B cell formation. Derived from stem cells in bone marrow. Acquire unique antigen binding receptors in the bone marrow. Receptors interact with just one antigen. So remember, the antigen is that kind of chemical message that is on the outside. It's almost like a name tag. Think of it. It helps to identify, protein message helps to identify what it is. And exposure to that antigen causes clonal selection, division of cells specific for that antigen. Okay, so clonal selection of B cells. Only B cells with that antigen receptor that matches the antigen is stimulated to divide. Mitosis yields many cells of that receptor. So if you see in this diagram here, you have the square, circle, and looks like B shape antibodies, B cells 1, 2, and 3. And when the antigen is attached to the specific antibody that responds to it, or specific B cell that responds to it, mitosis results in many of those B2, but none of the B1 and B3. This allows for your body to only produce what it needs, so it doesn't have to go through a bunch of extra work that it doesn't need. So antibody coding gene recombines as B cell matures, produces variable transcripts that are translated to produce receptor portion of the antibody molecule. Similar process produces variable T cell antigen receptors. So we see right here the transcription of specific ones, transcription and translation based on the response needed. So with immunological memory, memory cells specific for an antigen are quickly activated to divide upon subsequent exposure to that antigen. So your body has a certain memory to the different things that you've been exposed to. Lymph nodes filter antigens from body fluids. Macrophages, dendritic cells, B cell and T cells in the nodes, and spleen mount a defense. So your lymphatic system mounts a defense. Okay, you ever hear swollen lymph nodes, that type of thing when you get sick. Antibody-mediated response carried out by B cells. Targets are intercellular pathogens and toxins. Antibodies bind to target and mark for destructions by phagocytes and the complement. So we see the antibody structure right here. Antibody consists of 4 polypeptide chains. Certain parts of each chain are variable, impact the antigen specifically. Virgin B cells becomes antigen-presenting B cell. Helper T cell binds to antigen MHC complex on the B cell. Interleukins stimulate B cell division and differentiation. Effector cells secrete antibodies. Okay, so you have an antibody response. Here are the 5 classes of immunoglobulins: IgM, IgD, IgG, IgA, and lastly IgE. Okay, so I would know the 5 classes of immunoglobulins. Okay, we're not going to go too crazy on this particular test, but knowing the different immunoglobulins, and there's a review packet that goes over exactly what I'm expecting you to know on this. Cell-mediated response is carried out by T cells, stimulated by antigen-presenting macrophages. The main target is antigen-presenting body cells with intercellular pathogens or tumor cells. Now, when we talk about this organ rejection, when you have a organ transplant, cytotoxic T cells can contribute to rejection of transplanted tissues. They recognize a portion of the donor cells, MHC complex, as self, view a portion as foreign, and treat the combination as antigen MHC complex and attack donor cells. Immunotherapy, all body cells display fragments of proteins they are making bound to the MHC on their surface. In cancer cells, the fragment may be abnormal. Immunotherapy uses this cue to attempt to destroy cancer cells, so it can have your own body attack the cancer cells. Monoclonal antibodies, manufactured antibodies against tumor-specific antigens, first created by fusing antigen-producing B cells from mice with cells from B cell tumors, now made in genetically engineered cells. Lymphokine-activated killers. Lymphocytes are extracted from tumors. Extracted cells are exposed to lymphokine and interleukin. And large population of tumor-infiltrating activated lymphocytes is then re-injected into the patient. Therapeutic vaccines. Researchers are genetically engineering tumor antigens to make them more obvious to the immune system. Engineered antigen is used as a vaccine to activate the patient's immune system against the tumor antigen. Immunization, a process that promotes immunity. Active immunization, antigen-coating materials injected, confers long-lasting immunity. So active immunization would be you get a vaccine, okay? Passive, you're getting just the antibodies. So in active, your body produces the antibodies. You get a shot, the flu shot, whatever shot you get, and your body produces its own antibodies. Passive is used in things like rabies, where you don't have enough time for your body to build antibodies. So what they do is they directly inject antibodies into you, and that's passive. All right. Another way to get passive immunity is infants get passive immunity from their mother when they are nursing and also through the umbilical cord. Allergies are an immune reaction to a harmless substance. You're genetically predisposed to allergies. IgE responds to antigen by binding to mast cells and basophils. These cells secrete substance that causes symptoms. Okay, so allergies are an overreaction by your body. Remember we talked about the histamine reaction before. You take an antihistamine that blocks that histamine reaction, so you do not get that overreaction. If anyone's ever had an allergy test where they use the little injections into your arm, and then you see the inflammation based on your body's histamine response. Anaphylactic shock is a life-threatening allergic reaction caused by the release of histamine by many mast cells and basophils. Airways constrict, blood pressure drops as capillary permeability soars. And finally, autoimmune disorders. Immune system makes antibodies against self-antigens, so your body makes antibodies to attack yourself. Graves' disease, myasthenia gravis, and rheumatoid arthritis.